Okay, we're going to go over some Punnett square basics. In our demonstration here, capital B is for brown-eyed individuals, lowercase b is for blue-eyed individuals, and as kind of the cases indicate, the brown eye is dominant in the phenotype to the blue eyes. Okay, so what we do is we have a square. The male is always represented on across the top. And in our example here, he's got brown eyes, but with a little b. So we put gene for one of the chromosomes here and the other one here. For our female, and this could be people, gorillas, horses, doesn't matter as we're looking at this. So our female also has brown eyes, but with a big B and a little b. So we put this down across here, individual genes. And then what we call fill out the square. Okay, so we bring the male's gene down here, down to the two potentials. The same here with the little b. Just by custom, always do the male on the left and you fill that in first. And then the female across, so she's going to donate a big B gene here and a big B gene here, and then a little b gene here and a little b gene here. So genotypically, we have our offspring of the four potentials. We have a big B and a big B. We have a big B and a little b. Which order they're in does not matter in terms of how it's going to show up phenotype or physically. And then our little b is here. So phenotype-wise, so these are the three different genes, sets of genes we can see. So we have a 1 in 4 chance of being both dominant brown genes, but we have a 50-50 chance of being big B, little b, and we have a 1 in 4 chance of having blue eyes, so no big B in here. But that's the genotype. Phenotype-wise, this is brown eyes. This is brown eyes. And this individual has blue eyes. So phenotype-wise, where this is 25, in the genotype, 25, 50, and 25%, this is 25, still 25%, 50, and 25, but since these are both brown, 75% chance that we're going to have brown eyes and only a 1 in 4 or 25% chance of blue eyes. Okay? Now we're going to pause and do the pedigree because that's an important thing here. Okay, so now we're going to look at a pedigree chart to try and see if we can work backwards to see what the parents have as we look at genes. In this particular case, we're going to look at detached earlobes and attached earlobes, okay, represented most commonly in a pedigree chart like this, the recessive gene or phenotype, the recessive phenotype is always represented by a hollow circle or square. The circle is usually the female and the square is the male. In this case, we're talking about men and women, humans, but this could be any species or any characteristic we're looking to evaluate. In our case, we're looking at detached earlobes, which is dominant. We do know that, and that's going to be represented by filled in circles or squares. And the recessive is not filled in. Okay, a couple of things to always remember when we come to a pedigree chart. It's simplest to start with the recessive gene. Why? Because like we talked about earlier in the Punnett square, it only shows up 1/4 of the time, and that's only if both parents, even if they express the dominant gene, have that recessive gene as part of their genetic makeup. Okay, so for example, in this family here with 5 girls, we have 2 of the girls that have attached earlobes. Even though both the parents are detached, that does tell us that if we're using D and A as our example, that does tell us that both parents have D and A as their genes. Okay? So that means that this individual's parents, one has a D and one has an A. Okay? We don't know which one yet. We'll see if we can figure this out without looking at a picture to tell if it's detached or attached. Okay? So this second person here, male, has detached earlobes, but we don't have any offspring to compare. This girl next, it has attached, and both her children are attached. Oh, I didn't put her husband in here. He also has, attached. So we know now, if these are both attached, that means both parents had to at least have one attached gene. Okay? Because we need both genes for this child and this child and this child to show up with attached genes. Okay? Because if we do our Punnett square with the dad, if we don't know here, then we're just kind of working a Punnett square backwards with the pedigree chart. We do know we need we need at least one attached genotype for it to show up with attached earlobes. So that we know one of these has to come from the father and the other has to come from the mother. Okay? We also know that at least one of them has to have the dominant. We don't know which one. Again, you can have a picture, and that may help us. But we don't know which one. But we know somebody has to have this. So it could be dad or mom. Could it be both of them has it? Yes, and so they would both have detached earlobes. Again, a picture may indicate that. But can we figure it out from looking at our pedigree chart? So we would look at their children. We have 1, 2, 3, 4, 5, 6, 7 children. What percent of the children have attached earlobes, or the recessive phenotype, or the physical property? We have 1, 2, 3 that are attached, so that means 4 are detached. So with a small number, that's hard to tell because that's close to 50-50 of the 3 and 7, but it's also kind of close to 1 out of 4, because if this was just up to 8 and we had 3 and 5, that could be close to the 1 out of 4, or it could be close to, 3 out of 4, because like here, we should have a 50-50 chance of them being both boys and girls in the family, because this family kind of did. We have 1, 2, 3, 4 boys and 3 girls. This family had all 5 girls. That's like trying to flip a coin 5 times in a row and end up with heads every time. So we don't know for sure which of the parents has the dominant gene. We do know both of them have to have the recessive gene, and we do know at least one has to have the dominant gene. But let's take it a step further. Let's change the example a little bit. Let's say instead of like what we have here, let's say this is the pedigree chart we have, where of the 7 children, 6 have detached and only 1 has attached. Do we still know for certain? No, we don't know for certain which one has it, or if they both parents have the detached gene and just have detached ears. It's likely they both do, but we don't know for certain with that. Okay? So that helps. We have another little image up here of maybe the grandparents. We're talking about this family, and everything's kind of in reference to this line here. If we had some other information backwards, we might be able to determine that. But that's how we work a pedigree chart. You always start with the recessive so that you can share your chart with other people. You want to keep your recessive as hollow and you're filled in as the dominant gene as you're trying to figure this out. And this is one of the many things we will do in biology at Chesterton this year. Thank you very much.